All right, so that first stage was glycolysis. So we took the glucose and we split it. We lysed it and we created two new molecules called pyruvate molecules. Now, at this stage, it really depends on what's available. So if it's an aerobic process, meaning that there's oxygen present and available, we're going to go through cellular respiration. Okay, so cellular respiration is going to happen. So what's going to happen is that the pyruvate molecules are actually going to move into, so they're going to move into the mitochondria to further kind of metabolize. However, if there's no oxygen present, we call this an anaerobic process, or sometimes there's a little bit of oxygen but not enough, it's still considered anaerobic. And so what's going to happen is the pyruvate molecules are going to stay in that cytoplasm where they were, and they're going to go through fermentation and reduce to still provide some ATP for the cells, just not as much as it should. So your body can switch anaerobically often because it depends on what you're doing. Um, this is a rock climber, obviously climbing up the side of a cliff with not much holding him there, and he's using his muscles to do so. And so each one of those muscles that's holding him up is going to need to have oxygen to produce more energy because he can't let go, right? But another example I always give is thinking about somebody who's running a sprint, a very short amount of time, a burst of energy, and maybe they're even doing hurdles. And so that's even more up and down energy that's needed. You can breathe in oxygen as you're running, obviously, but a lot of that oxygen is going to go to supply things like the muscle of the heart to keep it pumping in an exerted way, especially since you're running so fast. But all those other muscles that are working, all the muscles that are working, because it's not just your leg muscles, it's your arms muscles because you're pumping, your core muscles, your back muscles, all of them, all of those cells and all of those muscles need ATP. And so it's going to usually, in an example like that, go through anaerobic. Right? We're not going to have oxygen present. So what's going to happen is we're going to create a burst of quick ATP molecules through fermentation. The problem is we only get two of them every time we use a glucose molecule. So it's not extremely effective, but it works in the moment for very short spurts of time, like something that would be that example of running hurdles and sprinting. This individual is still using anaerobic, believe it or not, at times, because again, all the cells of the muscles, let's say in his leg, are contracting right now because they're holding him on the side of the cliff. And again, it's not a short burst, but he's using a lot of glucose and making only a few ATP to keep those cells working. So let's say we're in an aerobic situation, and what's going to happen is we're going to move into the mitochondria because there's oxygen prevalent. So what's going to happen as we move into this mitochondria is kind of that secondary kind of preparatory stage where basically the pyruvate is going to join another enzyme, which we haven't talked about yet, which we call coenzyme A. That's what you need to know. No other fancy name. And when these two work together with a couple other processes here, they're going to make what we call an acetyl-CoA molecule. And that is the important piece to kind of move on. That's the product here of this preparatory phase that's going to continue the process. This is that we make these new molecules called acetyl-CoA molecules. And again, there are two of them. And they're going to move into the mitochondria further into that Krebs cycle, or also known as that citric acid cycle. And then eventually, so this was step two, no, it was really step three. This is kind of step two of the process for cellular respiration. And then the last process is the electron transport chain, which is going to help us create the ATP. So let's just look at a visual because I think everybody looks at things very different visually wise. Here it's telling us a location, so cytoplasm versus mitochondria. Here is glycolysis, so here again, glucose creating to pyruvate. Now, in order for the glycolysis to happen, we need to use some ATP molecules. We gotta like spend a little money to make a little money. If you've ever heard of that in business, you gotta spend a little money in order for you to make a little bit money back. So we gotta spend a little ATP to make a little ATP. And unfortunately, in glycolysis, we're not making much ATP in the end. We only net two ATP molecules. So we haven't really made very much in this whole glycolysis process. Now, if oxygen's present, we're going to continue in. So here's that kind of secondary stage that I said, that kind of preparatory stage where the 2-pyruvate are now going to become 2-acetyl-CoA enzymes. Now, if you look over here, we're kind of each time creating some coenzymes that I told you were important. So the NADHs and then FADH2s. And with that, every time we make one of these, we have electrons that are moving as well. Remember? So we have that reaction happening where the electrons are moving as well. The third stage, like I said, is either the Krebs cycle, the citric acid cycle, which is a cycle. Every acetyl-CoA goes around. We go down twice. Okay, now it's a cycle. There's a lot coming in, a lot going out. You're going to learn more about that necessarily in lecture versus here. You're finally going to see we're creating some products now. We have our CO2s, that are leaving when we go through this Krebs cycle phase. In the end of the Krebs cycle, we're not really getting much. We're getting about 2 ATP again for each turn. So at this point, we've really made 4 to 6 ATP, and we haven't really made very much at all. But as you can see, we still are moving our hydrogen ions, okay? We're still moving our electrons, and so that number is changing as we go through this cycle here. So this is the important piece. We need to create a chemical gradient is what's happening. And once we hit the electron transport chain, that's when we're going to create all this ATP. So if you look over here, you can see from this group coming from glycolysis, I got 4 to 6 ATP. From this electron and hydrogen movement breaking down the pyruvate, we got 6. From the citric acid cycle, again, we're getting 6 NADHs, but we're usually getting about 16 ATP, and then again 4 here. So we are starting to rack up the ATP finally because of these guys, nothing else that we've done. And again, these have been created because of these stages. So let's just talk about what's happening here. So the electron transport chain is basically that. So the mitochondria has a plasma membrane just like all other organelles, and they have these protein channels that are going to, again, move the electrons. Okay, so they're going to move the electrons. And what ends up happening is outside of the cell, we get an abundance of hydrogen ions. They're all hanging outside not the cell, the layer of the mitochondria. So if this is the outside of the mitochondria, this is the inside of the mitochondria. So what we're creating is this chemical gradient where we have a lot of hydrogen ions in a big area. Now, along this plasma membrane, we also have these units called ATP synthase. All right, and they look like they have kind of a turbine at the bottom, which means that it would spin. And inside here, we have a lot of ADP and phosphate molecules hanging around. And we know that those two molecules are going to, those two elements, atoms or molecules, are going to make ATP. So this is almost thinking about like diffusion. Like when we're talking about a molecule in diffusion, high concentrations don't want to be together. If there's a high concentration, they want to move to a low concentration. Like the example I may have given about diffusion, when you're all kind of cramped on an elevator together and you might be like, oh, this person's eating, that person didn't brush their teeth, that person has BO, like I need personal space, whatever the case may be, you want to get out of that elevator as soon as the door opens and kind of move to an area where there's less concentration of people together. And so the same thing's happening here. We have a bunch of hydrogen ions hanging on the outside of this plasma membrane, and they really want to get back in. So they do. They're able to shoot through this channel, and when they do, there's a force with them. And when they create this force, it kind of spins a turbine at the bottom, and it creates these ADP molecules and phosphate molecules to turn into ATP. Now, the amount of hydrogen ions that are out here, it depends on how many we have from these processes. That's why I said this was important that we acquired all of these. And again, including the electrons that are there. So they spin to go down into the cell and they create ATP molecules. Now, ultimately, the goal here is that we create between 32 to 34, possibly 36. It depends on the cells. It depends on how many hydrogen ions we have, how much ATP we're making, which is a lot more bang for our buck versus the 2 in the 2s. However, it's still not a very efficient process. The one thing that I talk about that ATP synthase kind of represents is a turbine. And I always give the example of most of us live near Holyoke. We obviously go to school at Holyoke. And if you've ever been down to the Connecticut River downtown Holyoke, you know there's a big, huge dam on the Connecticut River. Now, the dam is to hold back the current, but there are parts of the dam that are open so that the current can flow through there. And obviously, it's flowing really fast, especially at certain times of the year, especially in the spring when the snow has melted off of the mountains up in the northern areas. And so what they're doing actually there is Holyoke Gas and Electric has hydroelectric static electricity. And so what they're doing is they have these big turbines, kind of like our ATP synthase, which have this long, like, kind of pipe to it. And then it has like that circular wheel that might kind of have like little paddles on it. I am not a good artist here, etc. But what the job really is they have the water flowing through the opening in the dam, right? The little opening here, forcefully. And what happens is this turns. And because of that, it's creating energy, different type of energy. It's not ATP, but it's creating energy that people of Holyoke actually receive in surrounding cities. And they harness that energy there. So that's what's happening there. And again, I don't know if anyone's ever been there. They have the fish ladder there that's open in the spring as well. Sometimes people go down and visit. So this is basically the idea of cellular respiration. Fermentation is completely opposite. Remember, needed no oxygen or limited oxygen, so it's an anaerobic pathway. Basically, again, it's providing a burst of energy of ATP, but only 2 ATPs per glucose. So it's not extremely beneficial. It is, but not extremely. So we can have alcohol fermentation, which we'll talk about, and lactic acid fermentation is what we have and we create in our own bodies. Anybody ever work out, do things not worked out for a while, or pushed your body in a certain way? And you get that soreness the next day. What that is lactic acid buildup, which is, again, a byproduct of fermentation in your body. So that's how you know fermentation has occurred. But when we're talking about alcohol fermentation, we're taking some type of glucose or sugar. We might give it a fuel like yeast, and we get fermentation. So we get alcohol, ethanol, and we get carbon dioxide. And again, we can try monosaccharides and disaccharides with this. So obviously, a byproduct is alcohol. Not always has to be alcohol, but for example, we'll talk about this one. So our monosaccharides, again, could be glucose, fructose. It could be lactose, which is milk, all right, which is milk, lactose. Sometimes people are lactose intolerant, but this is like a milk sugar. Sucrose is a disaccharide. Sucrose is very hard for your body to break down. So if you look at food labels and there's sucrose in those labels, that is challenging for your body, taxing for them to break down. And then starch are much larger. So we're talking about a large polysaccharide, which would be very challenging to obviously break down to use. Now, here's an example of what we would have done in lab. We would have taken yeast, we would have put it with water, we would have given it a fuel source, so we would have tried glucose, we would have tried fructose, we would have tried sucrose, we probably would have done a starch as well. We would have put it in temperature environments like the body temperature, so 37 degrees Celsius. We may have heated one up, we may have cooled one down and put it in the fridge or the freezer, and we want to see what happens. So what happens here is this. Now, how do I know something has happened? The byproducts are alcohol. I'm not going to drink this. I don't want to know how much alcohol might be in there. And the other byproduct would be carbon dioxide. So because there are all these bubbles forming right here, I know that fermentation has occurred. And that's because the amount of bubbles that I can see. So that lets me know what's happening here. Now, if you make bread, you're going to use the same type of thing. You're going to have yeast. You're going to have some type of sugar source. You're going to have a few other things, right? You might have milk. You might have the flour and everything, and you put it in a warm environment. And what happens? The dough rises because, again, the carbon dioxide bubbles are within, and then you usually kind of have to punch it down, let it rise again. And again, when you bake it, it burns off the alcohol, so you don't necessarily get drunk by eating bread or whatever, or intoxicated by eating bread. But there's other things that you could be taking in that go through fermentation: cheeses, yogurts, kombucha, kimchi, types of fermented food like sauerkraut and pickles and carrots, but those you would find only in the refrigerated section, not down the canned aisle, not down the jarred aisle. But if you go into the produce section, section, and there's always stuff in the produce section that's cold. You might find salad dressings and things like that. You can find fermented food there. You can, at any store, you can buy kombucha now, kimchi. Real Pickles is a local company that does fermenting as well.